Hi dear students, good morning. I'm Dr. Mashur. Today we are going to discuss direct selection of recombinants. Okay, in our DNA technology, we are going to discuss direct selection of recombinants. So we have told that there are different techniques used for selection of clones. Okay, and this is the first technique, that is a direct selection of recombinants. Okay, direct selection of recombinants. Okay, in this procedure the desired clone only will be survive on the selection medium. Okay, in this procedure the desired clone only will be that means the required the clone of required gene only will be survive on the selection medium. Okay, to select the clone. Okay, to select for a cloned gene, it is necessary to place the transformants onto an agar medium on which the only desired recombinant can grow and others cannot grow. Okay, to able to select this for a cloned gene, we have to place the transformants on an agar medium. On that medium the desired clone can only able to grow and others cannot able to grow. Okay, and we can discuss this method by using a simple example. Okay, we can explain this method by using a simple example. So here you have to clone a antibiotic resistant gene. Okay, in this procedure we can amplify a specific antibiotic resistant gene into the another bacteria. We have to clone an antibiotic resistant gene on another bacterium. Here you can see one plasmid is there. Here you can see one plasmid is there. Okay, one plasmid, his name is R5 R65 plasmid is there. R65 plasmid is there. Okay, R65 plasmid is there, and that contain one gene, kanamycin resistant gene is there. Okay, kanamycin resistant gene is there, and this plasmid also contains some other antibiotic gene like kanamycin, chloramphenicol, streptomycin, and sulfonamide also present in this plasmid. But we have to clone this kanamycin gene only. Okay, we have to clone this kanamycin gene only. Okay, and we can see here, you see this kanamycin gene is present inside a EcoR1 site. Okay, this lines indicates the EcoR1 site. Okay, this line indicates the EcoR1 site. So we cut this plasmid. Okay, we have to identify, isolate we have to clone this kanamycin gene in the bacterium. So we'll cut this plasmid using EcoR1. Okay, we'll cut this plasmid using EcoR1. So that time we'll get around 13 different fragments. Okay, we'll get a 13 different fragments. Okay, but in that our desired fragment, kanamycin gene fragment, is present here, and that will clone. Okay, we cannot able to isolate this individual fragment from this mixture of fragments, so we have to clone this total 13 DNA fragments into the EcoR1 site of PBR322. Okay, EcoR1 site of PBR322, and we'll insert into the plasmid PBR322. Okay, here you will get around that 13 fragments transform that recombinant DNA here, and we'll transfer that recombinant, all these 13 fragments, into the bacteria. Okay, we'll transfer the E. coli bacteria, and after that we'll get, okay, we'll transfer this E. coli bacteria, and we'll grow that transformed E. coli bacteria in kanamycin containing agar medium. Okay, we'll transfer this that transformed E. coli bacteria into the we'll grow the transformed E. coli bacteria in the kanamycin containing medium. Okay, kanamycin containing medium. Okay, that bacteria, host bacteria, is susceptible to kanamycin, and this medium contain kanamycin. okay, that time we'll get only one colony. Okay, we'll get only one colony. That colony contain kanamycin resistant gene. That colony all contain kanamycin resistant gene, because we told that this medium contain the kanamycin. So other dual recombinants does not have the kanamycin gene, so that transformed bacteria cannot able to survive in the kanamycin contain, but this bacteria contain the this fragment kanamycin, so that colony is will be survived here. So we can directly select this colony from here. That's why it is called as a direct selection. Okay, this is the method of direct selection of recombinants. Okay, in the process of direct selection of recombinant DNA, that time the desired gene containing clone only will be survive in the selection medium. Okay, the medium that desired gene containing clone only will be survey. For that we have to plate the transformers on the selective medium, and that medium only allow that uh, desired DNA product to grow on that medium. Others cannot able to survive on that. For that we can explain this method by using a simple experiment. Okay, we can explain this that method by using a simple experiment. Here one that we can explain that by using a cloning of a antibiotic resistant gene cloning. Okay, here you can see one plasmid is there, R65 plasmid is there, that's contain one gene kanamycin, and that kanamycin is present inside the echo R1 fragment. Okay, echo R1 fragment. Here echo R1 site is there, echo R1. So when we cut here, they'll form a kanamycin free kanamycin fragment. But and this plasmid also contains some other antibiotic also is the chloramphenicol, streptomycin, sulfonamide is there. But we have to amplify this kanamycin. We have to clone this kanamycin. For that, we'll cut this plasmid with the echo R1. Okay, we'll cut with the echo R1. So we'll get a around 13 fragment. We'll get in that one fragment will be the fragment of kanamycin resistant gene. Okay, kanamycin resistant gene. So we'll clone this. We'll incorporate this fragment into the PBR392. Okay, we'll incorporate this fragment into PBR392 and transfer into the bacteria. Okay, transfer into bacteria, and that transformed bacteria will be allowed to grow in medium containing kanamycin. Okay, transformed bacteria is allowed to grow on the medium containing kanamycin, but only one clone will be survive here, because that only bacteria containing kanamycin will be survive here. Okay, only the bacteria that contain kanamycin resistant gene only will be survive here, because other that bacteria is a kanamycin susceptible, and that this recombinant also does not contain kanamycin. So that's this unwanted, this kanamycin absent bacteria cannot able to survive here. So we can directly select the recombinant kanamycin clones from here. This is the direct selection of recombinants. Okay, so thank you for listening my class. Thank you.